Hello, everyone. This is Berk from the Center for Engineering and Medicine at the Massachusetts General Hospital and the Harvard Medical School. I'd like to share some of our recent work on supercooling and more specifically the deep supercooling of water and its applications to cellular preservation. This is the work mostly performed by Dr. Heishui Huang, who was a postdoctoral fellow in our center and has recently moved to China as an associate professor at Jiangjiantong University. First of all, I'd like to address why we're interested in supercooling and why we think it can address some of the gaps in the biopreservation field. We all know about cold storage as a preservation modality, which is widely used around the world for organs and some cells. Cold storage is conducted above the melting point of water and usually at plus 4 Celsius to avoid freezing or phase changes. Timescale of the preservation offered is usually low and counted by hours to at most days. It's an easily applicable method to the preservation of pretty much any living matter. On the other end of the spectrum, we have cryopreservation with phase changes like controlled freezing and vitrification. These theoretically offer unlimited preservation time, but they also come with poor outcomes for some cells. Moreover, successful cryopreservation of organs and large tissues have eluded the scientific community despite recent advances to offer potential solutions in this arena. In this context, we at the Center for Engineering Medicine think that supercooled aqueous solutions can provide an optimum intermediate between cold storage and cryopreservation. Supercooling is the use of metastable states of aqueous solutions below their melting point where phase changes are avoided despite such low temperatures. While our center and others have shown that supercooled preservation around -4 to -6 degrees Celsius is possible for a few days, and even for large organs, we think that both the stability, temperature, and length of such supercooled preservation can be improved upon. We all know that the homogeneous freezing point of water is at around -40 degrees Celsius. However, achieving such temperatures without freezing or even achieving -20 degrees Celsius is exclusively possible when we use very small volumes of water as demonstrated by Biggs and others decades ago. This is especially true under normal conditions of pressure. Nevertheless, we also expect that theoretically we can achieve much longer preservation time via supercooled solutions if we could lower the temperature from our currently used ranges and improve the stability of especially larger volumes to increase the period of supercooling. To this end, our goal was and is, with our research programs at the center, to enable stable supercooling for temperatures below -10 degrees Celsius for large volumes, not just droplets, but large samples up to hundreds of milliliters, and long times, not just a day or two, but weeks to months. To address this goal, about 2 years ago, we stumbled upon a phenomenon which we now dub the deep supercooling. We think that with this approach, we can now achieve most of our goals listed up here to create alternative preservation methods for traditionally cold stored products like red blood cells and some exotic and hard to preserve cells like sporozoites. Okay, I will now stop laboring and explain how we achieved these goals, first by discussing our work on supercooling water down to -20 degrees Celsius for large volumes, and then I'll give a short discussion of an application on preservation of red blood cells. Deep supercooling of large volumes at low temperatures works by sealing off these aqueous solutions by water immiscible hydrocarbon phases in solid containers. Our hypothesis was that the ice nucleation probability for such a solution is likely the highest at the double liquid air and triple solid liquid air interfaces. Because of the much larger area of the liquid air interface, we thought that the contribution from this double interface would be the larger effect. We also thought, as a possible additional benefit, that the replacement of air by another fluid with a much higher viscosity would have the effect of dissipation of any disturbance at this interface. So I'll just walk you through the results of this experiment on the right hand side, where we have 1 milliliter of ultra pure water in polystyrene tubes, and they're sealed by oils like mineral oil, olive oil, and I'll highlight these for you, paraffin oil, and nutmeg oil. We kept these solutions at minus 13 degrees C for periods of up to 7 days and assessed their cumulative freezing probability. We found that all oils, with the exception of olive oil, suppress any ice nucleation for up to 7 days without any dependence on time and showing great stability, as you can see right here. Olive oil, whose freezing point is above minus 13 degrees C, curdled above the water, but still somewhat suppressed nucleation to some extent in a time-dependent fashion. As you can see, olive oil was curdled down here. Upon having these first results, we asked ourselves how much further can we push this phenomenon in terms of temperature and volume, and whether there's a volume dependence on the effects of sealing with these oils. We looked at volumes ranging from few microliters here to up to 10 milliliters here with mineral oil sealing, and saw that over the period of a day there was very little, if any, dependence on volume for freezing events. As you can see, this is a pretty steady result. Mineral oil was able to suppress nucleation at these range of volumes easily compared to non-sealed controls, which quickly froze. We then pushed these volumes further up to 100 milliliters and sealed water samples with both mineral oil and paraffin oil. This is where we start to see the effectiveness of mineral oil sealing breakdown, only suppressing freezing for half of the samples. Take a look at it here. At minus 13 degrees Celsius. Mineral oil sealing had almost no effect on freezing for these 100 milliliter samples when we pushed the temperature further down to minus 16 degrees Celsius. Paraffin oil, on the other hand, was extremely effective at suppressing freezing events for these large 100 milliliter samples at minus 13 degrees Celsius, and it showed no volume dependence at minus 13 degrees Celsius up until this 100 milliliter sample. At minus 16 degrees Celsius, however, even with the paraffin oil sealing, we observed that 40% of the samples froze. And we also conducted other large volume experiments at down to minus 20 degrees Celsius. We looked at hundreds milliliter samples at minus 20 degrees Celsius for 100 days, and 30 milliliter samples at minus 13 degrees C for 300 days. And an interesting observation we had was that at minus 20 degrees Celsius, we had 8 out of 35 samples survive without freezing. On day 3, these same samples all went to survive without freezing, also for the whole 100 days. We thus think that the freezing in our sealed samples are likely case specific and due to impurities rather than stochastic events. However, this point remains to be further investigated. Having established the sealing approach can work to dramatically reduce the freezing events in water, we wanted to examine whether there's a critical sealing where we can observe the full suppression, also to test our hypothesis about the effect of the interface. We thus created oil seals on the water phase using 5 different volumes of sealing oils. As you can see right here, we created different seals. The lowest volume we used was 0.01 milliliter. This only covered the triple solid liquid air interface and minimal the rest of the interface. 0.15 milliliter fully removed the air-liquid interface, and 3.5 milliliter was used to see if there's any effect of hydrostatic pressure. With these sealings, we then did a temperature sweep from -10 to -20 degrees Celsius using mineral oil as the sealing oil. We found that the ring seal, uh, as we called it, which just targets triple interface, um, has a very small effect in suppressing the freezing and only at the higher temperatures, as you can see here. The partial seal, which covered more of the partial seal here, I'm sorry, the partial seal, which covered more of the air liquid interface but still left some of the water exposed to air, had a much stronger effect, but it was not as effective as the other sealings, which covered 100% of the liquid surface, as you can see here. We thus concluded that complete removal of the air-liquid interface is the most important reason for the freezing suppression that we observe. Up until here, we relied on the surface sealing by oils, which are not well-defined mixtures, but they are known to contain a large fraction of alkanes. Accordingly, we used alkanes of varying chain lengths to assess the sealing effectiveness for up to a week at very low temperature of -20 degrees C. We also studied the corresponding short alcohols since we thought the hydrogen bonding of water molecules with the hydroxyl groups of alcohols can further stabilize the interfaces against disturbances beyond what we have seen earlier. We see in this graph for the alkanes and alcohols that the alcohols of very short chain length, as you can see here, are very effective in suppressing freezing events even at -20 degrees Celsius, but the effect is lower for longer chain lengths. Where I need to remind myself and everyone that monolayers of long alcohol chains, up to 28 and 32 carbons, are traditionally used to actually initiate ice nucleation. We saw an opposite trend with alkanes, where C11 had the greatest suppression of freezing, but the shorter chains, especially lower than C8, had very low effect on the freezing suppression. We discussed these results in much more detail along with what's happening at the interface in our paper from 2018. We also looked at the mechanical stability of these supercooled samples via vibration, ultracentrifuge, and thermal disturbances, and finally ultrasonication, where samples were first cooled to -20 degrees C. While all sealing approaches were almost 100% stable under vibration and thermal disturbances, only alcohols provided nearly perfect avoidance of freezing when we use the extreme disturbance of ultrasonication. This further confounds our thinking that alcohols through hydrogen bonding at the interface might be providing the highest stability for these supercooled samples. Before I conclude my talk, I want to show one application where we have applied this deep supercooling approach to cellular preservation. In what we still consider preliminary studies, we looked into supercooling of red blood cells for up to 100 days. We used two different preservation solutions at five different temperatures, including cold storage temperature of 4 degrees C. We found that while the commonly used solution for cold storage does not do at all well at the supercooled temperatures, the UW solution with travel supplementation did really well at the supercooled temperatures for up to 100 days. We then tried to quantify the quality of preservation in these experiments by assessing the hemoglobin recovery in these cells on day 42 and day 100 for both solutions at all temperatures. We showed that even without substantial optimization, the deep supercooling can enable higher quality preservation with significantly better hemoglobin recovery and morphological features at -13 and -16 degrees Celsius up to 100 days. With that, I'd like to leave the slide up here and thank all our funders for their graceful funding for this work. And I'd like to acknowledge my team members, especially the ones that conducted the work, like Dr. Huang and Mr. Camilo Ray Bedon. And I'd like to note that we also published a paper this year on the preservation of MSCs, in addition to the RBC work that I've shown here today. So with that, I thank you for your time and I welcome any questions.